So today, I would just like to introduce you to what the group's been up to over the past year and why we formed. So the definition, the MPI Manuka honey definition, has been implemented since February 2018. And just shortly after that time, we started to discuss ways in which MPI and industry could work together to discuss any issues we have with science, and particularly to do with Manuka honey. So the group was created shortly after this, and so today I'll just go over some of the science projects that we've been working on. So, as the name suggests, we're a collaboration between industry and MPI. And the logos to the side there just let you know who's involved and the representatives in the group. We formed to ensure that the Manuka honey regulatory definition remains fit for purpose going forward, particularly with any changes we might have in test methods and new science opportunities, and also give us the chance to recognise opportunities to develop industry value in any new science that gets developed. And so we're prioritising a range of different projects, as I mentioned, and the three of them just listed there are the ones I'll be discussing today. So we've got a stability study on the honey markers, we've got how we can understand the influence of sampling techniques, particularly on test results, and then finally it's a pilot trial and some of the findings for that to establish a New Zealand honey reference collection. So firstly, if you look at the stability study on the honey markers. So there's many stability studies have been done on honey markers, and they all usually have lots of different samples, and they usually come up with different conclusions as well. So this gave us a chance to work together and design something a little bit different to try and understand stability of honey markers. It's just in the planning phases, so I'll just share a few of our approaches just now. So first, if you think about it, you want to source honeys that have similar levels of one marker that you're interested in. For example, 3-phenylactic acid, one of the markers in the definition. You then want to find samples in two different categories. So the ones A in the red drums there would be samples that are greater than 400 milligrams per kilo. Then we have samples that are slightly different, so they've got values below the 400 milligrams per kilo. We then want to take those samples, and then we want to blend them together. So the idea is to try and create standard levels of the 3-phenylactic acid that we're interested in, and that means we've got a range of samples of the similar starting levels. So we then test that sample for both that marker and a range of other honey markers that we might be interested in. And then we store it for, say, up to 24 months, and we keep those samples at 3 different storage temperatures. We test them periodically throughout that time, and then we test them at the very end again, and the idea is to look at any patterns of change we might see. So do those markers stay the same over that time? Do they decrease? Do they increase? Or maybe they do a bit of both. So as I said, we're just trying to source samples at the moment to try and start this study. And we won't just focus on that one marker, we'll have to repeat the whole setup, but for a range of other markers. So the second project that we're looking at is about the influence of honey sampling techniques and how that affects any test results you get from those samples. So I know that was brought up yesterday in the session a little bit, so we're doing a bigger piece of work now on that. So sampling and the way you take a sample can really influence how confident you can be in the test results you get from that sample. So is it actually representative of the bigger batch, the bigger drum that you're taking the sample from? And there are many factors that can affect how good that sample might be. So when did you actually take the sample? How long has it been sitting there for? How have you stored the honey? How old is it? And under what conditions? What about the extraction process you used? Also the amount of honey that's produced and you're actually taking a sample from in the first place. And also the test you actually want to carry out on that sample. And so our aim for this project was to understand what can actually influence the collection of the honey samples and therefore how the test results might change with these. And we set up doing this by looking at different stages. So the first part of this project was actually to do an industry survey. And the idea of the survey was to basically say, well, what do you do now? How do you take a sample? When do you take the sample? How big a sample do you take? And just all those other factors that are already happening out there. We then used the results of those surveys to design a range of experiments. And then as part of those experiments, which are done on site, not in a lab, we'd have to then source samples that fit in with those experiments, and we have to test all those samples. And then it comes to analyse all the results and try and understand what they actually mean. And we hope to use all the results to develop a sampling guidance. So it's just to give you an idea of what might affect how good the sample you're taking, and also therefore the test results you get from that sample. So at the moment we're actually only at this stage of the process, so I'll just spend some time now talking about some of the results from the survey and what we're hoping to do in the next upcoming months. So some of the results to date from the industry survey, we went to 12 different production facilities. It doesn't seem like a lot, but we asked some standardised questions and we realised that it was actually getting the same answers to all those questions, so we were pretty confident we'd captured the variability out there. Generally, the importance of sampling was really well understood. Everyone did do it quite well, but there was a variety of techniques used, and it depends on the point in the supply chain that you're taking a sample from. The samples, as I said, were taken throughout the supply chain, but most people were really into sampling when it came from sampling the drums, sampling the IBCs, and also sampling the retail ready packs. So there were some challenges people did note with sampling though, particularly when it comes to using a coder or an auger. They said that actually they couldn't get all the way into the bottom of the drum. Sometimes it just got the top part, layers got mixed up and all sorts in between. And particularly when the honeys were stored for long times, or it was a low temperature, or really viscous types of honey. And there was also the obvious questions around, well, when should I take a sample when you're filling an order? How many samples do I take, and just when? And so from all those results, we've kind of focused on several main experiments going forward. So the first one we want to focus on is the final retail ready product. So this is about not how you homogenise or blend, it's actually how the drums and how they vary and what that means when they're blended together and what the final product might actually say. We then want to focus on the drums themselves. So as I said, there's over time the honey might layer and might stratify, and so we want to see about when you take a sample, how does that actually, do you just get the top, and is the top very different from the bottom and everywhere in between that drum? And then we'll just double check that any of the results we get from the drums and how variable they are, does it actually apply to the same when you're storing it in much larger amounts in the IBCs there. So the next stage for this project is we've visited one production site and got one set of sampling underway. I've not seen any results from that, so I'm afraid I can't share them with you. But we're hoping to go to repeat that at various production sites in the next couple of months. So the final project is looking at the New Zealand honey reference collection, and this was a pilot trial. So the idea is that this will be a partnership both between MPI and industry. MPI may use the reference collection for regulatory purposes, and industry can use it for a variety of other science projects and in-market research. And so when you think of a reference collection, I guess everyone still thinks of a library really as a true and trusted source of information. And so the idea is you can do the same with the honeys. So the idea is you'll have a honey reference collection that you can be confident in, so that any new science projects or any new science work is used on New Zealand honeys that we all trust and can rely on. But to help build this reference collection and to help plan for any future science work, there's lots of unanswered questions. For example, when and where was that honey collected? How was the honey collected? What even is that honey? How do you characterise it? And therefore, what test results are available along with that honey? And so our aim was to develop and trial basically a range of different protocols to help try and establish a New Zealand honey reference collection. So I'll just briefly go over the approach we took for this trial. So we have an apiary site there. We then get honey from that apiary site, and the question we want to answer is really, should this honey be in a New Zealand honey reference collection? So firstly, we had to look at, well, how do you actually collect a honey sample? So we had to develop these protocols to collect both from the hives and from the drums. And we wanted to know things like the location of the hives and where the honey was from, but also how big a sample could we get? Could we get several frames, or is it only 500 grams? And therefore, how do you then store that sample, package the sample, and then send it off to the lab? Because we're not just focusing on Manuka honey, we're talking about all types of honey reference collections. So we also did a botanical survey at each apiary site to see if we could quantify the different types of vegetation that were growing at that site. And then we also had to look at extraction and sub-sampling protocols. So you've got the honey or the frame now is in the lab. How do you get it out the frame? How do you have to filter it? How do you sub-sample? How long do you mix it for? A whole range of other questions we had to try and test out here. And therefore, how many sub-samples should you store and how large should those sub-samples be? And then of course it's around any test results. So we've got the honeys now, what else do we do with them? So what test results do you need if you want that honey to be part of your reference collection? So I literally just got the test results for some of our honeys this week, so I've not had a chance to look at them in detail, but I'll just quickly go over some of the botanical survey results. So we had about 107 apiaries for the pilot trial this year, and of those we had 18 apiary sites that we carried out a botanical survey, and that was across the 5 regions you can see listed there. Now we did a botanical survey by doing a number of different plots at that site, and the number depended on how diverse the vegetation was at the site. So if you think about, you've got an area of land, you've got some hives in there, and that's pretty uniform, not much difference. So you can randomly put your plots kind of as so to measure the different types of vegetation there. But if you get a scenario where it's a little bit different, so the different blocks there represent different vegetation types, and so you have to make sure that you randomly place your plots there, but make sure you capture that range of diversity of the vegetation. So from doing all these, we actually recorded the occurrence of 855 plant species there, and that was across the 57 plots that we got across the sites. So you can see there that although we've only got 18 apiary sites, it's quite a labour-intensive job, and quite a lot of plants were recorded. So the graph here just shows how frequent they were across the plots on the Y, and then along the bottom there you've got the different types of plant species. So if it is the next one, just highlights in blue some of the sort of ones we might expect. So you've got the Kunzia species with the most frequent there. Then you've got Trifolium repens, so the clover. You can see Lectospermum scoparium just in the middle there, and at the other end we've got quite a bit of gorse. So still a lot more to do with all this data, it's only just the initial phases. So the next steps of the pilot trial really are to try and make sense of all this information that we've collected. We've got to analyse the test results, of course, of the honeys, and then relate that to all the botanical data and all the traceability information we've collected. So you can see from the botanical survey, it is quite a lot of work, it's quite costly, it's quite hard to get around the area that a bee could possibly forage in. So we're looking at other ways to look at measuring the amount of vegetation there, for example, satellite images and GIS layers, and that might be another way to estimate the plant abundance. And nextly, we're going to actually collate all these results, lessons learned, what will we do going forward to try and plan a medium to long-term New Zealand honey reference collection. And of course, we then have to plan for the upcoming season to try and get more samples, more sites, and a better coverage across New Zealand. So that's me. I didn't do it all alone. There's lots of people to acknowledge and involved in all sorts of the projects, so big thanks to them. And I think now we're going to hand over to Tony, who's going to focus on some of the uses of the reference collection and why you might want it going forward. Thanks.